Hello, Rizanabe. Hello, Managard. Welcome, welcome. Sorry, I missed you. You missed the sausage stream. Hope it went well. It did go well. Yeah, you can always check out the VOD. It was not chaotic, but hectic. I would probably skip till about 3/4 the way through if you want to see the sausage making in action and the increase in chat. Talking about me making other voluntelling me to make other things in the future, and the increase of sausage based puns, because there's not a good way to make sausage without the inevitable I don't even know what to call it, suggestiveness of making sausages. Just the terms and the actions and the movements, everything. Hello, Brian. Connect to my... Come on, phone. Show up. Work. Yes, always allow. Allow. Come on, come on, come on. There you go. No audio, please. Haha. Ah, by the way, sneak preview. I decided that I am going to do another cooking stream someday. It's going to be this though. If I can get it in frame. I picked this up today at a store. I don't know if you've seen these before, but they are completely gummies, and they have all these instructions to make each individual part from little packets. Next time I do something cooking related, that's what it will be. All right, did I choose the wrong chord? What's going on? Okay. Did I get Google Fiber? Barely a lag. No, the hurricane prevented Google Fiber. So no Google Fiber, at least not yet. Google Fiber will be coming someday. Someday. Okay. Yeah, I don't know why there's no lag. Yeah. So I've been updating all the software that I use, which caused some initial problems, but I think it's sort of making things better. Are we looking at sausages under the microscope? No, but thank you for reminding me. I was gonna grab out a bit of the sausage casing to like put on right away. I'm actually gonna go grab that right now. Thank you for reminding me. Let me put on something. Maybe I can find something that's like alive to like look at for a bit. Well, for one, I didn't drag the equipment to the kitchen for today. Yeah. That took so long to set up. Put back together was fine. Next time, if there is a next time, it will go way easier, I think. It was harder. So getting it back up here was better than it was to set up down there, because if you saw the picture on Discord, it was kind of just haphazardly like on chairs and on top of board game boxes, right? There was that was tough to get all set up. Up here is just on my desk. And I took a picture of where all the cords were originally, so that might have made it a little bit easier on me. All right, I have a fantastic new lichen sample. I don't know if there's anything on it yet. Like, as in, I don't know if any of the tardigrades or other organisms have woken up quite yet, but I do want to take a look at it. This is, oh, it's so bright. And also so zoomed in. So the storm kicked a bunch of fresh lichen off of tons of trees. So of course I went around and collected all of them off the ground. Google Fiber will probably show up the day Twitch is able to beam video directly into our brains. For you, you assume? Google Fiber, I watched them lay the fiber down like 2 years ago in front of our house, and it took 2 years for them to actually be like, hey, we're here, you can have us now. I'm sure it was like some thing with utilities they had to work out. Tardigrades sleep. Yeah, tardigrades, like other, so they're not insects, they're not arthropods, but they are close to them. Like other members of the Ecdysozoa is what they're called. They undergo periods of rest. Now, it's not like sleep like we think of it, but more likely the tardigrades that are probably on this sample, but I just don't have them, like, I'm not finding them quite yet. They're probably in their ton state, which is where they are dehydrated and undergo cryptobiosis, where for all intents and purposes they die. Like, there is no metabolic activity, at least as far as I'm aware. But upon rehydration, they pop back to life. That's something right there, but what are you? Oh, there's a tardigrade. Beautiful. Oh, 2 tardigrades. I think there's one on the top right, too. All right. I'm going to leave this here. Maybe we'll see some tardigrades pop out. Oh, there's like a mite right there, too. So we've got a mite up there on the top left and a tardigrade at the bottom right. I'm going to leave this bit of lichen under the microscope, and I'm going to go grab a piece of that that sausage casing, which is intestine, and we can look at it there. You know, the mite scared away the tardigrade. Well, maybe it'll come back into frame. Anyways, I will be right back. Whoops, I meant to do this. Hello, David. Sorry, and also hello, Micro. I was just off getting some pig intestine / sausage casing, and somewhere on this little piece of lichen there is a tardigrade, but it's hiding from that mite. Is that what mites normally eat? Lichen? I actually don't know. I don't know what type of mite this is. It very well could be. Well, it's been covered. I don't know. I would assume a mite wouldn't scoff at a tasty wet lichen meal. All right. So I don't actually know how this is going to look, / you know, how to get the best look at this. So I'm going to grab a little bit of this. It's also fairly gross. I'm trying to be good and touch it with only my gloved hands. Because remember, this is intestine, more or less. I'll get kind of a thin part. I will use my little scissors here to cut it. And I will try to, what's the word? Flatten it? Ooh, it's very... that does not want to cut very easily. Which I guess is why it's used for sausage, because it's quite sturdy, despite the fact that it's quite thin. Hello, Boomer Knight. We are looking at a tardigrade, but also a mite. So the mite is the big thing that's floating around up here, but there's that tardigrade on the left-hand side just peeking out. Yeah, far more vegan-friendly stream, although I did just grab out a bit of sausage casing that we're going to look at under the microscope. It's tissue, though. It's tissue. It's just tissue. And I won't pull it up on screen. We'll only look at it under the scope. And I don't even know what we'll see, because it is quite thick. I need something sharper. I need my X-Acto knife to use as a scalpel. You have an art project for me. Shape some lichen in the form of the Empire State Building that allow the mite to walk all over it and destroy it. The mite does look quite monstrous. Yeah, it's huge compared to that poor tardigrade who's just probably trying to go about her day. And this giant mite keeps moving her lichen around. And for all I know, mites eat tardigrades. I don't know. I've tried to look this up, like sort of the food web of the microscopic world. There's... I must just not know how to search for that scientific literature very effectively. Because it's kind of like there's the main things that like everyone knows about apparently. Like this or that organism eating this or that type of like algae. But then when it comes to like, well, do mites eat tardigrades? Do copepods eat tardigrades? Do tardigrades eat other tardigrades? I can never really find good information. Probably because they're hard to observe, being microscopic. All right. That is so rude. Okay. So I'm going to try and lay this as flat as I can. I hope I didn't manhandle it too much. It's not dangerous, but I am still wearing the gloves, just because I don't want to have to get up and wash my hands. Immediately, at least. Okay. Nice and flat. Actually, that flattens out quite nicely. Do I have water? I'm going to put a little bit of water to keep it hydrated. Honestly, they don't even have great surveys about what occupies each trophic level in certain aquatic ecosystems. Yeah, I kind of assume that most of the food web is still kind of unknown to us. Yeah, the mite is very determined. Let's recenter this. Oh, beautiful little tardigrade though. That's a small one. I want the mite to go away, though. Okay, we will come back to this. I don't know if we'll be able to find the same one. But we will definitely come back to that. Well, as long as the mite stays there, I guess. Oh, a species of tardigrade that can eat other tardigrades. That's exciting. Ooh. Okay, okay. I'm trying to find a thin patch. So this Melnesium means little predators. Cute. And see, that's the other thing. I just kind of assume that most things at the microscopic level, like, why wouldn't they just eat everything else? Oh, oh, R mean little predators. Oh. Little predator is a cute name, though, if we can figure out the Latin for that. All right. So this is on a dish, but it's splayed out as best as I can. So this is the bit of pig intestine. It's primarily just collagen, most likely. I don't know. I assume in the process of preserving it, there's no cells left. Let's see. How can I smooth this out? I don't know if I want to put a cover slip on the top. Predator should be Latin already. Even if it's Latin in origin, scientific names tend to be even more Latin, or more fake Latin / Greek. So I would assume it would just be a little bit more exaggerated. Mod check cover slips. Well, I just want to know if putting a cover slip on would get rid of those bubbles. Let's zoom in. Oh, ooh, actually, look at that. So we can really see the what I believe are probably collagen fibers right there. You know what? I don't know what these little dark spots are. I don't know what's left after these have been sort of prepared for use in sausage. Let's zoom in a bit. I'm not really going to see much detail, except that you can really nicely see the cross-hatching of the fibers. Maybe what I could do is I could put a little bit of stain on it. The stain should only stain for nuclei. There's not going to be any nuclei left, but I might as well just try it out. I mean, it's here. Why not? It's pretty, though. I want to take a picture. There we go. It's on Discord now. If I should advertise this more, if you aren't aware, when I say I'm going to take a picture, I actually am taking a screenshot, and it's posted directly to Discord. Whoops. I believe I added just too much just now. It didn't work? What? Oh, Boomer Knight, I see that you just joined. Why is my microscope not working? And it hasn't worked since... Well, it worked last time. There must have been some update that I messed with. Music slightly too loud. Alright. I need to move some of this dye, this stain. All right, and that didn't stain anything. You now have a soda. Congratulations. There is a animal with the scientific name for small predator, Microraptor. Oh yeah, I know Microraptor. Raptor just means predator? That's cool, actually. All right, that stain did not stain much. Is that a nucleus? Nuclei, please. Not sure what that is. Microraptor. I guess that makes sense. I guess I could have guessed micro was going to mean small. You know, I'd really... Did I say nucleus? Is that what I said? As if it isn't obvious, I don't listen to what's coming out of my mouth when I'm streaming, so... because I was about to say nuclei, but then I realized there was probably only one. So this, whatever this is, this got stained a little bit more. All right, cover slip? Cover slip? I don't know if a cover slip will work in this setup. As in, I'm kind of just, so it's on a dish, but the dish is more thin than a normal slide, but then the sample is super thick. So will I be able to go full magnification? I don't know, but I'll try. I'm going to guess no, but we're going to try. I don't like how this looks like a close-up of skin with nasty-looking moles. Well, I mean, it's not... I mean, it's an epidermis, right? Not epidermis, but it's a epithelial tissue. Because I believe... Right? Am I getting that right? Because it's the outer side of the small intestine, which I believe is epithelium. So... Science! Welcome. Welcome. We're looking at some hog casing, which is highly processed, you know, pig intestine. I was just trying to remember whether or not the outer layer of the intestine was just epithelial tissue, because I believe that's just what we're looking at. It's very, very processed. And what these little dark spots are, the moles, as you said, Micro, I'm not exactly sure what those are. See, if I can... Okay, this is not going to work. Moving to the highest magnification is not going to work in this setup, but I'm willing to try. And my camera is a little off already. We're just going to have to deal with it. Yeah, sausage was great. I actually didn't try one yet. I was gonna have one for dinner tonight, but I did try the filling by itself, which tasted great. So that means there probably wasn't much of a change after I put it into the actual casing. But I'm doing well. How are you doing today? Yeah, I don't think this is... Oh, hey, look at that. Kind of lucked out there. Very, very, very close to the edge of what we'd be able to do. It's also not that fantastic. It's very fuzzy, but we can kind of see the striations of what I believe are collagen fibers. Doing good. Tired, but good. Yeah. That's how it goes. I mean, speaking of which, today is probably going to be an abbreviated stream. I already started late because I had to put my computer back together. All the villi even. See, I don't think the villi remain. I believe that layer of the intestine is removed. Oh, Google says it's the submucosa. I'm not even a fan of that view. Yeah, I'm gonna do a quick Google search. I don't actually know, what for sausage. What makes it through the processing? Yeah, exactly. Let me scoot over here. Do some Googling off screen. So what is sausage casing? How is it made? I mean, Wikipedia has to know, right? Origin processing. Okay. Blah, blah, blah. So it breaks the inner mucosa and washes that out. Natural casings are produced and sold almost exclusively by the hank, which is a unit of measure that is 91 meters long. Whoa, wow. Okay, well, apparently I bought like a hank or a half hank or something like that. You've only ever harvested cows. You had 91 meters. It is very long. I don't think I had a full hank. Okay. Yeah, it's not really. Oh yeah, submucosa of the small intestine. That's what you're saying. Okay. Okay, so the epithelium is removed. I see. Okay, wait. Let's quickly go to here. So mucosa is where the villi are. Apparently that's where the epithelium is. And then you have circular muscle on the outside that helps with the, what's it called? That movement, the peristalsis. And then the thing in the middle, which is, I think, primarily just collagen, the submucosa. So that's what was taken. So honestly, we've kind of seen all we need to see from this, right? We've seen the collagen fibers, and that's apparently the majority of what's there. I'm fine with that. That's pretty cool. Okay. I am going to put this away and put it somewhere so I don't forget about it, so my cats don't come and try to eat it. That would be very gross and possibly dangerous, since there's a methylene blue on there, which is staining the 0 nuclei that are on there. It looks like my camera's dislodging. It did not like the transport to the kitchen. All right, we'll fix that. So I do want to do some tardigrade collecting today for that ongoing project. I'm effectively just drying them in the same spot. So this is my little tardigrade house right now. It is a small ring, a small square of 3D printed material to act as a sort of barrier. And then in the center is where I've been dropping tardigrades and allowing them to dry out. Unless I find a whole bunch in one sitting, that's probably going to be the easiest way for me to deal with them. So I will do some tardigrade hunting today because I got that sweet, sweet hurricane lichen. But I believe I will start off by just looking at this ongoing culture that I have from the lake. There has been some movement, as in some evolution of what this looks like, and I have not checked on it in a couple days, so I'm curious. And I'm looking around because I'm looking for my dish. Yeah, it's my hurricane lichen. I mean, there were so many good pieces. I felt very strange. I mean, I don't have it here, but I had like a hunk like the size of my forearm off of a tree that I found on campus, and I felt like such a goofball walking around with this piece of lichen. And of course, it was the day before a football game, so they were setting up. So there were a lot more people on campus to look at me strangely as I walked by. I was like preparing myself to get stopped by a cop and have to say like, "Oh, don't worry, I'm a scientist. This definitely isn't suspicious in any way." I was taking my pet lichen out for a walk. Nothing wrong with that. Yeah. Biology people are always carrying weird stuff around. Why are we like this? In undergrad, I was brewing some things. I was doing an experiment to see if I could. So I like brewing beer very rarely, cider more often, but still like once a year, because honestly I can't drink that much of it, or we can't. But in college, I was doing a lot more experimentation with fermentation. Experimentation with fermentation. So I was doing things like I was trying to, like, you could buy like amylase enzyme. So I was like taking like pumpkin extract or pumpkin puree and mixing it with amylase and trying to ferment it to see if I could like break up fermentable sugars, or the like starches into fermentable sugars in the pumpkin puree, and then ferment it. I was doing that in like these weird jugs and stuff like that. And I put them in my closet. And well, not just my closet, but next to my closet was like a utility closet, which was nice and a stable, warm temperature. And apparently while I was gone one day, our landlord came to show the apartment around, and he was showing some people some things, and he opened that door and it was like a bunch of like weird buckets and like flasks and things like that with like the little fermenter stops on them. And apparently he was very confused and had to ask my roommates, and they're like, "Oh, don't worry, our roommate's a biology major." And he apparently accepted that as a totally logical, like, "Yep." Weirdest thing you've ever carried around is probably a human skull. For how long? And how did you carry it? Did you feel like you had to be very careful with it? I've never had to like move a human skull around. As a scientist, I'm least likely to accidentally produce methanol. Actually, normal fermentation does produce methanol. It's just that when you distill things, you accidentally might distill methanol if you're not too careful. It's all a concentration thing. The treatment for methanol poisoning is in fact just consuming more ethanol. So because it outcompetes ethanol or methanol in parts of your body is how I'll put it. So that's why like distilling is more dangerous. You can never ferment and then drink enough volume to have a lethal amount of methanol. So you've never understood what cider is. What is it? So cider is, so it's different depending on where you are. So, because I know in Britain it's something else. And in America, it's like evolving into two things. So there's hard cider and normal cider. Normal cider is just non-fermented apple juice. Non-fermented spiced apple juice, I think. And then, but cider as a class of fermentable drink is basically fruit wine. I mean, never tell a wine person that wine is just grape cider, but it kind of is. It's a sweet fruit-based wine, often fizzy. So what you do is, like for apple cider, you grind up a bunch of apples, get the juice, like real, like, I mean, you can take like a big sack of apples and grind them and squeeze them and press them, and all the, like, you'll get a couple, gallons of juice, but the actual physical dry bit of apple is probably like a small sphere. It's a crazy amount of juice. You get that, add some more sugar maybe, throw in yeast, it will ferment. That's cider, right? You can do that with lots of different types of fruit as long as they have fermentable sugars. I've heard about those biology folks. Is it normal for a landlord to show your apartment to people? You've never heard of that before. Where I have always lived, yes, viewing an apartment has always been like important. And since I, at the time, was living in a college town, in a college apartment, it's like we would move in August, and they would start showing the apartment in like October, like nearly a full year ahead of time. It was ridiculous. It was in a box. You took it out to school for your job as a student science outreach. Awesome. Hey, Rye Cerrone, which I meant to ask yesterday. Is that a pun on rice-a-roni? Just wondering. Yeah, you're like one of two people who discovered me cooking. Although I already said this, but next time I cook, it's going to be this. It's one of these Japanese candy maker things where you make all these things are gummies. That's the next time I'll do it. Not Chef Marcus. Drink some methadol, get to the hospital after a 1 week bender. Just rehydrate with a sodium chloride aqueous infusion. You're Rice-A-Roni, the San Francisco trick, the drag queen. Ah, you're a drag queen, and that's your stage name. That's such a good one. That's such a good one. Rice-A-Roni. That's a fantastic, wow. That's good, that's good, that's good. What, I'm not going to be making those nice sounding dishes from the Star Trek cards. I did mention that I might make, I could try that disgusting coffee syrup stuff that stupid Neelix was offering. See, that's the problem with those though. So they're all like, oh, Neelix's recipes. If Neelix were a real person, I don't think I'd eat his food, which makes, giving out his recipes on little cards a problem. All right. Enough talking, more microscoping for me. All right, so this is a this is a lake culture that was kind of boring last time I checked it, but then last time I checked it on stream, but then something happened and an explosion of life occurred, but now it's like crashing. Whoa, oh my god. Explosion of life is still happening. What is going on? Okay, this is gonna have to settle for a moment so we can actually see. There are a few awesome Star Trek cookbooks. Yeah, I have seen them. I've definitely seen them, and I'm curious. I'm curious. My curiosity has been piqued. I just feel like those specific cards from Neelix didn't sound too appetizing. If I want to cook some Star Trek stuff, it's got to be gach. Well, the best gach is raw, so I think you just have to. He only gave away the shit recipes, or all of his recipes were shit recipes. That is I feel like not even a hot take. Or maybe it's just because I listen way too much to the Greatest Generation podcast, where they just assume Neelix's food is disgusting. Ah, look. Oh. Yeah, look at all these friends zooming around. So these are... So at this level of magnification, these are not bacteria. So there are 100% bacteria zooming around in this sample, but not at this level of magnification. I'm a molecular biologist, not a microbiologist. So I, in my very untrained brain, would call the things floating around now at ciliates. Oh, except for that big thing that just flew by. We'll zoom in a little bit. So there are definitely also copepods in here, which I believe are arthropods. There's some Daphnia, which are freshwater crustaceans too. So I saw both. They're quite big though, so now they're going to just appear as, oh, look at you. They're just going to appear as dark splotches as they go by. Ah, look at this little friend. Look at him suck up everything. So I don't know if this is a ciliate or a type of rotifer. But you can see, if I can follow him correctly, he is sucking up things. Oh, look at him go. Siphoning through whatever this material is. I've seen this before, but I don't know what these are. if I set up an Amazon wish list with them, you will buy me a cookbook just to watch me cook some. Oh no, I don't want to convert to a cooking stream. Also, I don't want you to buy me anything. I'll buy it myself. Because that does sound fun. Some Bajoran Hosperat or some that's good. These are some eukaryotic homies. Absolutely. Yeah, so bacteria are prokaryotic. And they're generally small in size. Oh, look at him go. Look at his... Oh, wow. Look at that. You can see his little cilia if I stop banging the table, filtering through all this good food. It's like playing Spore, but for real. I'd love to play Spore on stream if it didn't just crash randomly by itself when you're not streaming. Lab work is just cooking we shouldn't eat. Absolutely. That is, yeah. Oh, look, he's making hella current. Go, little guy. Are we seeing it eat and poop at the same time? Yes, but also no. Like, it is probably expelling waste. But this little guy, if he is indeed a cilia or like a rotifer, he is not going to have a traditional mouth to anus digestive system that we have. But he's also probably expelling waste. We just can't tell. Or at least I can't tell. I'm going to try to take a microscope shot again. He's missing a lot of the stuff as he's pulling it towards him too. Yeah. So the stuff that you're seeing like shoot by is probably just stuff that's just kind of going by, not making it into its mouth. Wow, look at that go though. He is making a lot of current. Mad current. I need to do this.
Fascinating.
That's how I feel about this. Suck. I'm laughing and I'm causing the table to wobble. Oh, he looks so good though. Okay, I'm gonna try to just... I'm glad he kind of calmed down too. He kind of stopped moving. Because these are swimmers.
Fascinating.
Oh, he got a big piece of something. I really hope to see my favorite... are they rotifers? Ciliates?
Fascinating.
I don't know. My favorite slurper, Stentor. They were in this lake before. I just haven't seen any in this. Yeah, it's just really fun to watch. I absolutely agree, Rye. I also like his little, if you look at the back, like little back flagella thing. Redeem, name it, Suck. Suck 3000. That's what his yeah, that's his name. Suck 3000. Where's font size? Select font. There we go. Stentors are their own family, you believe? I trust you. Oh, does the microscope light not drive the sample? Yes, it absolutely does. This microscope is very old. It's older than me. Honestly, probably older or just as old as other people on stream. It gets extremely hot. There is quite a bit of liquid in this sample, though. So they will heat up. They will absolutely heat up. But the water should be good for a while. Nothing is older than you. I don't know how old you are, so I probably have something in here older than older than you. I mean, though this microscope is a pretty good contention. I'm not even gonna guess how old you are. I'm just kind of guessing like what is old. What is old? I don't know. Nah, the tattoos. Where'd they go? My Chakotay tattoos. My problematic Chakotay tattoos. But these are only like 25 years old. I'm older than these tattoos. There's still not much space for them to move around or away from the camera, right? So, there is about... How big is that? I don't know my metric. Give me a second. So these... Whoa, another one. So he has... Oh, he left. So they have about 4 millimeters in height, depth, up and down to go. And then they have 60... 30, wait, pi, what's 30 squared? They have about 60, like the diameter of the dish is about 60 millimeters. So they actually have quite a bit of room to move, you know, up, down, left, and right, and also X or the Z axis. All right, he left. Goodbye, suck 3000. All right, what are we doing here? From the looks of me, you have underwear older than me. Oh no. Your microscope is older than yours. Yes. You did tell me you had a very old microscope. I remember that. You have a microscope with a cover that says "Made in West Germany." It's reasonably old. Yeah, whatever my microscope is, it was made in China, and it was made sometime in like the 1970s. Okay, you're also older than the tattoos. 140 million year ammonite fossil. You have a couple fossils downstairs. He was run off by Suck 4000. Today you learned 1-cell creatures are jerk faces. 30 times 30 is 900. I probably could have done that in my head. And then 27. So approximately, what is that? 2,700 millimeters squared? That doesn't seem right. All right. He and his current disappeared. You're so old in high school. You sat next to John Microscope, inventor of the microscope, heir to the microscope fortune. All right. We lost him. But things have probably settled. Oh, look who's peeking out. Under this little bit of plant matter. All right, okay. Yeah, I think we're gonna be able to find lots of these. So we're gonna go look around a little bit, just see what else we have here. We have a lot of these smaller ciliates. Zoom out a little bit more. That's why does this look so bad? Why are you looking so bad? White balance. There we go. That's much better. Gotta remind me to change my camera settings. John Microscope invented the microscope when he tried to look at something twice at once. Hello. What is this? What are you? Okay, that's all that shaking's from me. What are you? Not really moving on its own. Wolfram Alpha says, okay, 100 millimeters. It's like a little cartoon tree. I'm wondering if that's, oh, actually, so whoever asked about bacteria, if you look very carefully, the little flecks swimming around, I'd be much more okay saying that those might be bacteria, because look how small they are in comparison to everything else we've been looking at. But I wonder if this is like the fruiting body of like a fungus or something that's like right there. Sorry this is so shaky. This is the problem of no cover slip, is every shake or every little bit of movement is translated into the sample, especially if it's like free floating. All right, let's look around. I was, for a moment, I thought that was going to be a rotifer because they tend to attach themselves, or some types of them attach to the slide like that, or to the slide, to substrate. We can see a lot of things. Oh, hey, look who it is. I think. Two of them. So both of these are rotifers. So you can see they're not stentor. I don't think they are, because they got little propellers rather than like a ring or a C shape. Although they might be. What do I know? Yeah, so these are both of these rotifers. Oh, you know what? That other one might have just been a rotifer that was just kind of waiting around. You can see how they have attachment points, and they, like on the bottom, like they have a little stalk body, and they attach themselves to this bit of food down at the bottom. Oh, they aren't rotifers either? Are you sure about that? Oh, sorry. Oh, they look single-celled. Okay, let me look it up. You know, someone who actually... Oh, you're right. What am I thinking of? Maybe these are stentor of some kind. You're absolutely correct. No, they're still not. Vorticella? No, you're right. Vorticella. All Miss Frizzles got it. Yeah, because they're much more bulbousy body and then that little stalk. So that other one we saw was absolutely Vorticella too. Why hasn't anyone created an app where you can just take a picture of the microscopic thing and the app identifies it? You know, maybe that exists, but I don't know. Wow, look at them go. Oh, they're like tag teaming this little vortex that they're making. They were one of the organisms that Van Leeuwenhoek named animalcules. Oh, interesting. That's fine. I knew who you meant. Wow. The one on the left is like main vortex, and the one on the right is kind of like sucking them in. There's not enough... You think there's not enough data to make AI capable of identifying microorganisms. I don't know if I agree or disagree. I just wonder if they could just sort of make a broad stroke, like, tell me, hey, this is not a rotifer. It's Vorticella. But what type of Vorticella? Who knows? Wow, look at them go. Oh, you totally need a drop of color in the water to see the vortex? That's a good idea. That's super tough, though. I mean, they themselves are in an area far, far, far smaller than a drop of dye. How can I do that, though? Okay. Also, that one is off on the plane. I'm just going to do a little bit of a checking quick on what sort of scale we're looking at. The only thing I have is food coloring, and I'm not sure if that's toxic to these guys. And I honestly think diffusion might cause the color to happen a lot quicker. I'm willing to try it, but I don't think it's going to work. A lot of species really only can be told using specific stains or gene sequencing. That makes sense. Absolutely. So it should be possible to make an app to make broad stroke identification, but there might not be a market for it. I suppose we'll have to wait for it. Yeah, see, that's what I would like. You just broad stroke, like, hey, I have a microscope, but I'm also an idiot when it comes to microbiology. Just tell me what broad classification we're in. Food coloring is toxic to humans, so you think it'll be to them. Well, okay, we'll let them live then. Yes, it's a bacteria. See, that's all I need. How do these creatures control their cilia and such? They don't have nervous systems, right? No, so they don't have nervous systems at all, but you don't need nervous systems to control things. So cilia, so I don't know these specific types of cilia. And I forget if it's flagella or cilia. But basically, if you just have a sort of coordinated set of proteins, all you need to do is activate them in sort of a cyclical fashion, and suddenly you've got them going all like this. Add in a couple, 100 millions of years of evolution, and you can get really tight control over that. Let's see. So yeah, flagella is a propeller. Cilia are beating back and forth. Yeah, cell signaling. I mean, yeah. So they're not thinking like, hey, I need to do this. They're responding to signals from their environment, external and internal signals as well. Whether or not they need to continue to feed or it's time to stop because conditions on the outside aren't as good or something like that. The more gregarious one. I said the E word. Environment? Cilia are wonderful. Shared a lab with a group investigating them. They do so many things. Yeah, they do. That's basically how you function when you wander into the kitchen. The one on the right looks like a lizard's eyeball. That is an interesting thought. Let's zoom in on it, if I can control my microscope. Evolution. Ah, I see. Not really been able to, can't really get a good look at him. Oh, yeah, there's tons of bacteria playing around with whatever thing they're attached to. I'm pointing at my screen like you can see it. I bet that's a piece of just like rotting plant matter. Oh, he just got bonked by someone. All right. Wonderful rotifers. Not rotifers. Vorticella. Vorticella. Microscope shot? Why aren't you working, microscope? Oh. I need a digital laser pointer. So I actually have this. This just little arrow that, oh, whoops. This little arrow that I can do. Like this one's my favorite. Can you see it moving around before I let go? I assume so. That one's my favorite. This one is trucking along. I could try updating my video drivers to get Spore working. Okay. I unironically like Spore. And I've played it off and on since it came out in like what, 2011? 2009? I don't know. I feel like for years I've been updating my video drivers. I think it's just a unfortunately incompleted game. Okay, you can see it move. Awesome. Okay. So These guys are super fun. Oh, so they are attached to the thing, or they're feeding off of it, or maybe feeding it. Okay, so yeah. Whoa, whoa. Oh. Little bit of an incursion there. So you can see how when they get sort of, for lack of a better word, scared, they contract like that to protect themselves. Okay, so their mouth is at the top of the trumpet of the triangle. They're sucking things in. We can see lots of particles moving. They probably aren't eating those bigger ones. They're aiming for smaller things. The little thing that attaches them down to a substrate, that isn't a feeding mechanism. That just keeps them in one spot. They can detach it freely and move away if they want to. You don't think you could play the later Empire parts of Spore now that you've played Stellaris? Yeah, it was fun at the time, but so many better things have come out. You think you will do what they are doing? Go feed on organic matter? Fair. Yeah. Go get some lunch or early dinner. Lunch, dinner, or wherever you are. Okay. I am going to move along and see who else is around. Wow, this lake sample, last week it was asleep. Today it's just going nuts. Oh my gosh. Oh, there is a party going on. What is happening? So I can't even really oh my God. I can't focus on this because they're all going in and out at such different levels. Don't know what these are. Why are there so many in this one spot is my question. Okay, I'm gonna have to probably leave them alone because I don't think I'll ever get any one of them in focus unless we move to a cover slip. What is this? I don't know. I don't know what he is. Because he has cilia on the front. I wonder if it's an unattached Vorticella. Or something. Where are they all in a rush to? They got a meeting? I think so. Oh. Actually, yeah. I think that is a Vorticella. After it got bonked by that other guy, it kind of like squished down. Yeah, I think he's just stuck or something. Sad. Very sad. Wow, look at them all go. What's this big gloopy thing? So now the trick for me is going to be keeping this culture alive. Don't tell my significant other this, but I did look into buying an aquarium to keep lake water alive. I don't think I'll do that, but you know, I need to figure out like what to feed it. Like I've seen like you can feed these things milk and just little bits of plant matter every once in a while. What's this? And why can't I focus on it? Oh, you're actually trying to figure out how to mod Hive Worlds to not remove your lovely event planetary features while you watch this. What are you talking about? Oh, Distant Worlds Universe? I like games like that. I'm gonna write that one down. I don't think I've heard of that. Distant Worlds. Where is it? Distant. Oh, Stellaris. Oh, I've heard of Stellaris. I was trying to look at whatever this was, but I can't seem to. I'm at the edge of my slide. Or my stage, I mean. And you're waiting for a tentacle to smash through? Yeah, I don't know what that is. It's I think it's alive. It's a cell. That's what it is. There's something moving just off camera. I think. Oh yeah, it's not one of those guys. I still would love to see a stentor. So I will also, what I can do is at some point I will go sort of more broadly, like zoom out actually for once, and we'll see if we can look at any of the Daphnia or copepods that are in here. They're definitely hanging out somewhere. They're just we're not in the right plane to see them. Let's see. No stentors. No anything, actually. I am tempted to take at least some of this and put it under a cover slip to see if we can get a much, much closer look at anyone who comes along for the ride. Like you. Take another look at one of these guys. Oh. Come on. There's another, what I think is detached Vorticella. You need a GIF of any police procedural saying enhanced to instantly picture zooms in and gets clearer. I bet I could do something like that. I could at least integrate like an enhance chat command or something like that, or channel reward. So by the way, a lot of the channel point rewards right now are sort of being tested out in terms of like if they aren't too annoying, I'm going to change them into chat commands so that they're free. But for now, they're behind the channel point paywall. Ooh. I can't actually see. This is close to enhance. Except it's dehance. Oh. I did hydrate, by the way. Thank you for that. Yeah, the view screen one is fun. There's another version of the view screen which I feel like has to be, I could add, but would be very, very rare. Like I'd only allow it once, probably per stream. It's basically like a 30-second long clip of a view screen, green screen. So that's where I would show up, and then it goes back to characters from Star Trek looking at each other, you know, apprehensively because of what they're seeing on the view screen. And Hans, a verb to add people named Hans. And Hans. By the way, you're also a friend of DeSoto. Awesome. Yeah. Long time listener here. We've got a couple people who have popped on because every once in a while I'll advertise on the Discord. I feel like there's a lot of overlap. Mostly because I talk about Star Trek a lot, especially on Wednesdays. My one regret is I have not been able to see Greatest Generation live in person. I was waiting for years for them to show up in my area, and of course, after I left the area and moved 19 hours away, then they decided to come by. So, oops. All right. I am not seeing much new. Uh-oh. Except, hello. We've interrupted something right here. Ah, stupid controls. Seems like we've got some mating going on. But I'm also at the very edge of the plate, which is distorting the water, making it harder to actually focus. You started over from episode 1. It's so different from what it is now. Yeah, I listened to an early episode like within the past couple of months because a friend of mine started listening, and I was just very nostalgic. Yeah, it's fun getting to hear where the bits and clips come from. Can we get some cheesy music playing? Yes. If I don't lose them. Ah! Oh, that scared the shit out of me. I don't know if that works. It doesn't freeze this one. I forgot I need to fix that so it actually freezes the frame. It only works on the main streaming one. That was also loud for me. I lost them. They left. Okay, I'm gonna zoom out though. I want to see if we can find any copepods and or Daphnia. So I'm gonna zoom out a bit. Okay. They realized we were watching, so they ran off to get a hotel room. A tiny, tiny hotel room. Or an Airbnb with outrageous cleaning fees. All right. So I know, whoa. What is with this? Look at this. Like, why in this particular area? I don't know what these are. Okay, you know what? These are going to go under the microscope. I'm at the end of my slide, which is how scientists say they're irritated with someone. That's a good one. Okay. Speaking of my slides, where are my slides? Okay, I'm going to grab some of these. I think I might put on some methylene blue, which will hopefully actually kill them so they stop moving. But under the slide, we'll be able to view them much more easily. So, because I really want to check out what these are. Are they kind of moving? Are they swarming away? Okay, my slide is going to have a little bit of extra detritus on it, unfortunately. We're just going to have to deal with that. I just got a Discord notification that I was live. Is my Discord not actually doing anything on time? Oh, they left. Here they are. Here they are. Did I get them? It was on time when you got it? Okay. I just noticed that the other day. That I was like getting notifications after I stopped streaming. So I was worried that other people were also getting that. I don't know. Between getting like a new phone recently and updating all this software, which clearly killed off my screenshot taking abilities, I was worried that other things were broken. I got them. Thank you. Move them off the slide. Okay, slide time. Please ignore the dirt that I left on there. They are not dead. You've noticed that sometimes Discord will renew a post notification hours after it actually posted. Oh, okay. So I add a little bit of methylene blue, which stains eukaryotic nuclei. It also kills cells. So these guys must just not be too susceptible to it. Unfortunate. But we'll zoom in because we'll have a much nicer view of them. Yep, my phone just buzzed again. Look at them go. Oh, they're not going to slow down for me, though. Unless that one does. I think that's one of them, right? Going to zoom way in before it's too late. Oh, look at them. Okay, what do we have here? Oh. All right, so I can just barely make out the cilia. Oh, and it's gone. Well. Found him. They really raven. Oh my gosh. I really need to fix my stage, my microscope stage. It's so bad. All right, so I'm at the highest magnification, which is 40x plus the 16x of the eyepiece. This is some old plant. Cool. By old plant, I mean old algae. And I can't see anything because everything's way too small and too fast. Okay. Let's go back. I think what I'm going to grab a little bit more. I'm going to dump this sample back. And then just grab more. I think I might grab sort of more. If I'm careful, I think I can kind of grab from around the perimeter where I've noticed a lot of like little worms hanging out. I don't know if they're flatworms or what they are, but they were not microscopic in that I could just tell that they were there with the naked eye. And I'll also try to grab more just off the surface, like substrate. Okay, let's see how that works out. So this is same sample, just a new bit of it. I didn't see any I didn't see those copepods that I thought I saw before. A little unfortunate. Yeah, because I'd really hoped to see the flatworms that I saw before. I'm very interested in them. Or again, what I personally, me, would call flatworms. Or even those Daphnia or copepods. Some of them. Yeah, whatever these like little ciliates are, they are just everywhere. What is this? Also, are those all Vorticella? Yes, they are. Oh my god. It's Vorticella City. Look at them all. Oh no, I can't actually.
Fascinating.
Okay, I'm gonna screw this up, I think. Oh, there they are. I just want to move this ever so slightly, but I have to move it with my hands, so that might not be good. There we go. Although I'm still at the end. Okay, hold on. That's close enough. Yeah, so we got 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 off to the side. What are they all doing here? What is this? Oh, there's even more on whatever this is. Interesting. So they have a preference in what they're actually attaching to. I wonder if this is like a piece of rotting plant matter or something. That's so cool. I'll zoom in. Oh, look at them go. Look at those little propellers. Now I'm tempted to put them under a cover slip. And by tempted, I mean I'm going to do exactly that. Yes. Where are my microscope slides? I'll get a fresh one. And a fresh cover slip. Okay, I got to zoom out though to triangulate. So I can get this piece of whatever. Look at them. Looks like something a cat coughed up. Maybe I have 2 cats. And they've been trying to drink the water that's inside this. They have fresh water. And they want this cup of, of pond water that I have on my desk. It's because it's on my desk. Because they love to steal stuff from my desk. The cup has more seasoning than fresh water. Ew, gross. I mean, you're probably right, but also gross. Whoops. I think I got it. Just need a bit of extra water, though. And probably some sand or something to make sure it's not crushed. All right, okay. Let's see if I actually did get it or if that was an illusion. Cover slips, cover slips. I want to see my little Vorticellas. Oh, there's going to be a bubble probably right on top of them. No. Or they're perfectly fine. Yes. Look at that. No, it didn't get blown away. I was using a pipette to suck them up. Sorry, I didn't say that before, but that's what I was doing. There we go. Let's find okay. Let's find one that's oh. Got some that are on the move. The unfortunate thing is my 40X is a little dirty and or scratched. They're kind of waving in the wind. This one, well, this one's pretty stable. Oh, wow. No. You stupid stage. Now you want a microscope for Christmas? Yeah, you can get good microscopes like fairly cheaply. Oh, we just watched that one open up. I just have so far been a little too cheap to actually invest in a better one. I really should. OpenSet has a good microscope that I looked up once and is a very good price. You can get them for like $400 or $500, and they're perfectly workable. They're probably better quality than this one, but about the same style. Except the newer ones have LED lights and also will have are like a tripod. So they have two for the eyes and a third up in the top. And what that lets you do is like look and then also put a camera up, which is something I cannot do. I'm kind of doing this, an arm's length away at my screen. Oh, these. Okay. I'm going to zoom back out just one. So I want to take a look at them wholesale. There's so many of them. I really want to know what they've like parked themselves on. Looks like one of them's trying to get in on the action. So what is this? I can't really tell, but I feel like those threads might be some kind of fungus. So yeah, I'm just gonna guess it's some kind of, plant matter. I wonder if that's like a technique to increase the chance that they get something delicious. If they park themselves on something that is being actively degraded by bacteria, then maybe that increases their chance of sucking up something like bacteria. At least That would make sense to me. Yeah, so what is Vorticella? If I search Vorticella diet. Vorticella diet. Bacteria and small protozoans. There we go. They also reproduce by diffusion. Diffusion. Wait, no they don't. Wait, how do they do this? Oh yeah, it looks like they can asexually reproduce. Huh. You lost 30 pounds in 4 weeks on the Vorticella diet? Ugh. Can you imagine just eating spoonfuls of just bacteria and protozoa? You know, I bet we could market that diet. Like many things, I feel like we could market something that sounds like that because it sounds scientific. Make a bunch of money, but it's unethical. Aw, got one all by himself. All right, well, while we're here, okay, I'm very happy with that. But while we're here with this cover slip, might as well take a quick little sneaky peek around. Hello, what is this in the rocks right here? Volvox, is that what it's called? Yeah, maybe Volvox, I don't know. New Maybelline anti-aging serum. Now with 40% more Vorticella than other leading brands. That suggests other leading brands already have a noticeable amount of Vorticella. Maybe she's born with it. Maybe it's microorganisms. Again, like we dig up some, shit paper on microorganisms as like de-aging, whatever. Slap a label on it. I think we've got marketing gold right there. Unethical, but marketing gold. Woo. I actually thought that one was broken. Ooh, that did put the yellow light straight in my eyes, though. It was a slightly darker color before. That's fine. I need to, that reminds me, oh, we're back here. I need to program in one. I like the light baby blue plus red. That's a really pretty light combinations. It's like true cyan and red. But I need to program that in. They already claim that chlorella can cure all kinds of things, so I don't see why we can't use another algae. Vampire bordello? I do not understand. What did I just search? Bordello of Vampire Pleasures? Facebook group? What is this? What have I stumbled on? Should be a color option for spooky month. Ah. Well, what two colors would you say that is? Or one color? Because you can do something like this. Or not. Oh, right. My own command. Spooky. A deep red, almost purple. if you have the hex code, so not deep enough. Coming to the stream late to immediately hear Vampire Bordello. You would know what Vampire Bordello is. Resident vampire expert. So deep red purple hex. Okay, let's try this one. Color red and this. I feel like it's just not showing up on the light quite as well as I might want. Ooh. I like these color palettes. Okay, I'm trying some things. Hold on. I don't think my own chat will, or my own bot will kick me out, or not kick me out, but prevent me from doing too many of these. Okay, I feel like I need to face them a little bit more. They kind of just diffuse to a white. Oh my gosh. Yeah, big raid. Wow. Dr. WD40, thank you with your raid of 88. Welcome everybody. You caught us just playing with my light situation here. That fact lady, thank you for the follow. Oh, now I feel like I gotta actually go back to the microscope. Welcome everybody. My name is Dr. N, but you can just call me Marcus. I'm a molecular biologist. I like to play around with microscopes and talk about science fiction. Today's more obviously a microscope day, but we've also gotten into other shenanigans. We have this wonderful 1Z followed by many underscores. Thank you for the follow. Right about here, we've got a wonderful little Vorticella group. So we're just checking out some wonderful lake samples today. And I was going to go look for a tardigrade at some point, and except for this, Vorticella is doing a little dance right now for us. So let's zoom in there. Meanwhile, my chat is also playing with my colored lights. Okay. Yeah, you caught me playing with my lights. We need to find a tardigrade so we can name it. All right. Well, first of all, look at this little friend. Dracos4, thank you for following. Yeah, I really appreciate the raid. Always fun to have more people watching. Oh, wow. With the just like dirt and bacteria in this, you can really see like the little vortexes that he's making. It's just so pretty. Look at that. And they're so efficient. I just honestly, it blows my mind. I'm sure it has to do with like the viscosity of water at that level, at that level, but it doesn't seem like it should be working that way. You vote for Henry for this one, or Henry for the tardigrade that we still need to go out and find. Regardless, yeah, this one, what is he doing? Oh, he stopped spinning. Okay. So you want a tardigrade, we can find you a tardigrade. Okay. Goodbye, Vorticellas. I think the other Vorticellas, they picked up and left. You want to name this one Yuri, after Yuri Grigorovich, famous ballet dancer, because it keeps spinning. That's a good name. Look at him go. Oh, yeah, you're playing with the colors. Yeah, you gave me quite a red color, but a very muted red color. That's the unfortunate thing. You can do any hex code for the colors, 1 to 2, and it will do it on both. But it kind of doesn't, not all the colors work, unfortunately, in that they just don't look the same as if you look at them up on Google. Okay, let's find a tardigrade. Goodbye, Vorticellas. I appreciate you. Any clue how long it takes them to do the stalks? Like to make their little attachment points? I actually don't know. I hadn't seen them. I've seen them detach, and they detach very quickly, but I haven't seen them actually like attach themselves. I've seen them bonk into each other trying to attach. James Gurney, welcome. Hello. Any word on my 30x coverage? So did I share what is with all these mites? Oh yeah, thank you, Brian. Yes. If somehow you're following me and not Dr. WD40, you need to go follow him. Amazing streams. I very, very, very often pop in and watch and or listen for just hours. This is really cool. Well, thank you. It is just pond water with my extremely out-of-date microscope, but you know, the microscopic world is super fun. This sample is lichen, by the way. It's my hurricane lichen brought to me by the recent hurricane. And I was hoping to find some fresh tardigrades in here, which we did find some before. We just got to get lucky and find another one. Like, not that one, not that one. Everything is interesting if you look closely enough. Absolutely. Oh, with the lights, it's probably best to start with purest colors and work your way down from there. Is that some kind of virus what we are looking at or something else? So I think I know what you were referring to. It was kind of like a little spiky boy that I went by. No, not a virus. Viruses are far too small to see via light microscopy like this. If it was a spiky thing, I think that's a type of fungus that I've seen before. Or you saw the mites. So should we call the tardigrade Ian? I mean, you know, argue amongst yourselves about what you want to call the tardigrade once we find it. You had an ex named Ian. Hard no. Oh yeah. Here's a spiky thing. No, this is I believe this is the start of a fungus. I see these very often in these freshwater samples. Just I haven't seen what they turn into. Where's? Okay, we literally saw a tardigrade before. Where are you? Stop hiding from us. I feel like, oh, also, thank you for the follow. Let's see, Hi Head Jester. It's a little hard to read from this far away from the screen. So I hope I didn't miss any letters. Okay, I do want to zoom in really quick. Okay, it's not a tardigrade, not a tardigrade. But these lichen samples are really fun once they start growing a little bit. So, like this. I believe what we were looking at is a type of algae. I don't know if this is necessarily still true, that people think that lichen is like an algae bacteria symbiote or something like that. But there's certainly bits of algae that hang along in lichen. And I don't know what this is, but these are definitely little strands of individual algal strands that are like growing outside. Okay, we're gonna have to name this tardigrade Waldo. Exactly. Waldo, the tardigrade who I cannot find. Yeah, see, we it's weird because for once we actually know, like this is a tardigrade verified sample, but then we went and looked at things that weren't tardigrades for a little bit. So yeah, so Micro, I feel like I saw some like Pop Science article, which of course I skimmed and didn't look up the sources, that we finally decided that lichen wasn't just algae and fungi. I'm sure that was wrong, or I misread it, or I'm misremembering it. Okay, we might go to max. By max, I mean minimum. Whoa. Oh, that's very blue. I think you found the bluest color. Come on. Why is this my camera's off. Or something. This is the purest blue. Okay, I got a mite right there. We got all these mites everywhere. Oh, this is so blue. Oh. Oh my gosh. I'm going to turn these away from me a little bit. Thank you, the Matrix. It even shows on the microscope. Oh yeah, it would. If I had a problem with the lights though, I wouldn't allow chat to control it. But ooh. Oh, that's weird. The camera shows it like a cyan, but it's very green on my end. That's not how we spell color in this house, Micro. Also, I know what that color is because I've done that before. Try again. There we go. Pure white. The least exciting. Where? Come on. Oh, there we go. Thank you for the follow, Jelunik. Now I'm lit purely by the microscope. Well, and the two screens in front of me, but you know. Like the scientists of old, keeping warm by the light of my halogen bulb. Trying to find these stupid tardigrades? Come on. You know, they're so gregarious when I am here alone looking around on my microscope. But you know, I go on stream and they all hide. You get these ugly mites instead. What is this one? This one's like a light pink. They know I'm planning to name it and they're hiding from you. Absolutely. I feel bad. I'm skipping by some like interesting things. Yeah, this is like a pink color. It's oh, it actually looks kind of nice behind me. Yeah, it's yeah. It's almost as if my cheap cheap camera doesn't quite pick up color perfectly. It doesn't have the highest fidelity for colors. So it makes some a little bit more interesting, a little bit less interesting. It's a very flattering tone. Oh, good. The Matrix, it didn't seem to like that one. There should be something that tells you that it's oh, look what we found. Oh, all alone, all by herself. Wonderful. Color M cherry. So this is, for everyone who is new, a tardigrade. They are an 8-legged, also my emotes are all tardigrades. So tardigrades are generally, I think they're all 8-legged, microscopic organisms. They're animals, so they are multicellular. They have their fun little legs. They have a little face right in front. They are part of Ecdysozoa, which is kind of like a super, I don't know what the correct taxonomic term, but like a super family. So if we have like insects, so it's insects, right? Then arthropods are like insects and crustaceans, like crabs and such. Go one step further up, and we get Ecdysozoa, where we have worms, we have arthropods, and we also have things like tardigrades. You've never seen a live one before? Yeah. You know, I'm sort of collecting them. I'm trying to start a culture so that we get. Oh. Yoshi-io. Thank you for the follow. Yeah, so I'm trying to get like a little culture of them going. I had one going for a while, but they all kind of died, unfortunately. But we'd get a much better view if I put her under a cover slip, which unfortunately is a death sentence, because I can't get them back out successfully. Are tardigrades still using cell signaling? Absolutely. I honestly don't really know of organisms that don't use some sort of cell signaling. Although, you know, if James, you're still here, you can tell me whether or not quorum sensing counts as cell signaling. But, you know, absolutely, they are multicellular organisms, so those cells absolutely need to talk to each other. And are these water bears? Yes, they are. These are water bears. For a second, you thought I was going to say I'm collecting them because I'm trying to start a cult. Well, nope, a culture, but you know. Wait, did I say died? Did I say died? So okay. So tardigrades are, they're not hard to kill, but you are correct that they are hardy. They can go into something called a ton state, which is a state of basically death. It's called cryptobiosis. So all cellular functions stop, as far as I'm aware. Someone can correct me if like there's something that still happens, but they can basically stay dried out for many, many years just fine, just waiting for water to show up. But, you know, I could, oh yeah, because I said died, because I could, you know, squish them. Well, yeah, so I could, if I had a little, you know, manipulators, I could sit here and rip it in half. That would kill it. That's sort of a misunderstanding, is that because they can survive for many years desiccated, that suddenly means that they're like invulnerable. They're actually still very vulnerable. They're poor little microscopic little guys. But yeah. Okay. Let me see. Where does it come from, and who are we can destroy it? So this is from a lichen sample, and we can kill it so many ways, but I don't want to. They sent them to the moon. I don't know if that's true. They might have sent them up onto the, you know, onto the space station to do some experiments. I already have a cult, and it's you. Your prayers are you changing the lights. That's how I know that you think you're thinking of me. That I'm thinking of you. So maybe quorum sensing counts. You don't know. It depends on your definition. Well, my definition is that they're cells and they're signaling. So there we go. So that can piss off the quorum sensing and cell signaling people. Two fields I don't deal with. So not basically death, actual death. Okay. See, it's another definition thing. I just don't know if there was enough of any type of metabolic thing that happened that allows them to be technically alive still. I can't name it, or I can't kill it right after you named it. That would be horrible. So have we decided this one's name is Waldo, right? Let me just go ahead and change this, because we don't need Suck 3000 anymore. Waldo. Oh, very cute. Leave Waldo alone. Are tardigrades social with other tardigrades? Oh, that's a good question. I don't know. I don't know about that. They reproduce, well, actually, some of them reproduce sexually. So they have to, you know, come together eventually. And also like social, like they're not social. So like social insects, we think of like bees and ants. Those have very particular social structures. Other insects more or less only interact when they're forced to, / they go through reproduction. I would just kind of guess that tardigrades are similar to that. Not to the moon, but they were put on the outside of the ISS. Yeah. Send them to the moon to do experiments. They're cheaper than postdocs. Yeah, but I feel like they wouldn't repeat the experiments properly. Oh, you spent the points to name it Managrade. Oh, I'm sorry. I missed that. I'm so sorry. You are not that. You are Managrade. There you go. Do you feel like this tardigrade sometimes? Struggling in a watery void, unable to find any substrate to latch onto? I do. Thank you, Managard. The definition of cryptobiosis is zero metabolism. Good to know. Yeah, so no, Managard spent the points, so we're officially naming it Managrade. They will mate, but they aren't really social. Nice. Did I know that most bee species are not social? Yeah, I did. I just assumed that most people think of honeybees when they think of bees. Oh, Hazel X, which is 1, 2, 3, 4, 5, 6, 5/6 the name of my cat, Hazel. Thank you for the follow. Oh yeah, chat got really busy, so I did miss it. I have the activity feed. It's just that, you know, I don't always have time to double check that I didn't miss something. Bumblebees are better than honeybees. Fight you? I would not fight you for that because I agree. However, are you aware of carpenter bees? Carpenter bees where the males do not have any stinger, and yet what they love to do is the males will guard their nests and just bonk into things like other male carpenter bees. Any way to tag it so that it can be recognized if others are found? Unfortunately, no, except that I could take her out of the solution, put her on a slide, and let her dry out, and then we will have a preserved Managrade until we decide to rehydrate it. It's really moving like much bigger creatures. That's so fascinating. And what's the chain to the left? So that chain to the left is a chain of algae that either was a part of the lichen or was just on the lichen. And so it's growing in this solution. So they're growing out linearly. And yeah, they move around like bigger creatures because they're not basically bigger creatures. What am I trying to say? So they have the same class of genes that humans do that determine like our head is up here, our arms are here, our legs are down here. They have the same patterning genes as us, more or less, to make it super, super, super simplified. Should you be worried about this? No, absolutely not. I mean, these are everywhere. They do not infect people with anything. They just hang out and have a good time. They live on moss and lichen. They're just kind of around. Carpenter bees suck tennis racket practice before they destroyed your house. Okay, they do a little bit of house destruction. That is true, but I think they're cute. No male bees have stingers. They're modified ovipositors. Oh. Bumblebees are those really big ones. They're scary AF. Carpenter bees are a tiny bit bigger, at least in my area. You'd be okay with Managrade getting tunned? Okay, we can ton this tardigrade. 6 limbs. So they actually have 8 limbs. So right at their butt, they have 2 more limbs. So let me try and point it out right here. So right here, these little things pointing out right there are 2 more limbs at the back. So they have 6 plus the other 2 at the end. Yeah, 6 normal, 2 backwards. Okay. I'm sorry to have mentioned carpenter bees. It seems like there's some contentious feelings about carpenter bees. Okay. Yeah, let's let's dehydrate managrade. All right. I was born for this. Sort of. Okay. Let's put her on. Okay, let's put her on the tardigrade culture. Growing tardigrade culture. Might be a little tough to do properly, but I want to try it out. This is way too much liquid. If you see an insect as big as your thumb, you're burning your whole house down. I don't know what climate you live in, but, you know, depending on the climate, that could be like you're burning your house down every single day. You're wasting a lot of money in, you know, insurance and repairs. Okay. Okay. So what I'm going to do is I'm going to take my pipette. I'm going to take my partially 3D printed slide, little house for tardigrades. I will attempt to grab her and move her. So I've got to zoom out just a tiny bit. Just so I can see what I'm doing. I also don't know why that's black right there. I think I'm getting some movement with my camera. Okay. So we'll move her onto the slide, then we'll take a look and see if we can watch her go into cryptobiosis. Die, but reversibly. Reversibly die. Okay, I just need to clean off this tip a little bit. So, like, very asleep. Yes. The deepest sleep, but it's reversible for them. Okay. Okay, I'm going to try very quickly. Actually, I actually need to turn my lights something else. There we go. Okay, I think I got her. Sorry, yes, this was very intense. I think I got her, because you saw the tip come in and then she disappeared, right? I'm trying to add as little water as possible. Okay, I have to expel more water, I think, unfortunately. Okay, this is very much not ideal. I unfortunately had to take quite a bit of water. Although, there she is. Okay, so I did get her. Can I get some of this excess water off, though? That's kind of my question. Sorry, I'm looking over at chat, but I'm like very slowly trying to take off some of the excess water without sucking her back up. Otherwise, I'll just have to repeat the process. Because if I don't suck up enough of it, it's going to take forever for her to dry out. Okay, we're looking good. Water is getting pretty low. If she just heads for one of those edges, I think she'll, she'll get up there quickly. Oh yeah, thank you, Micro. You're making a good point. Let's see. She is very, very small, and she is in only 60 microliters of water, but still, just because of the magnification, it looks like a huge amount. Ooh, that was yeah, because that was tricky. She's still there. She's still there. All right. So now so what I've done now is I've increased the amount of light that is on the slide itself. You can actually just about make out the edges before I moved it drying up. Yeah, so you can just about tell that it's starting to dry a little bit. I might have to go in again to kind of soak up a little extra, but we'll kind of let her be like that. From what has happened before on stream is like this happens fairly quickly once the drying begins. She's so small and a little distressed. All right, Micro, you're saying they're so tiny they don't need any special organs to breathe despite being an animal. Yeah, they just get oxygen from the water through their skin. Okay, this will seem a little horrifying, but believe me, this has happened to her multiple times in her life already. They grow on things that are prone to just drying out. We live in a region that goes through very dry and very wet stages. So she's just going to go to sleep, and then she's going to be woken back up someday. Okay, where's she moving? She's moving towards the edge. Okay. I'm going to take the tiniest bit of paper. You monster. She's fine. She's fine. James, we gotta get David to tell us whether or not he wants to talk more about biotech more. I'd really love to, but I gotta get something on the schedule if I'm gonna make the time. Not that I wouldn't make the time, I just need to know. Yeah, I know he had COVID. I hope he's not. I know he's like ostensibly better. I just hope he's not feeling too bad. It responds that quickly no wonder they survive the process. It preserves before trauma occurs to the cells. Actually, so trauma does occur to the cells, interestingly. When you rehydrate them, the amount of time it takes for them to basically pop back to life is proportional to the amount of damage that was done to their cells. So she will incur a slight bit of damage, but it's nothing she can't fix later on. Oh, she's escaping. Okay, I'm going to soak up some of this water with a tiny bit of paper. I am going to zoom out, though. I really don't want to suck her up onto a bit of paper. That would be so anticlimactic. Uh-oh. Okay. Okay. They regenerate so many awesome longevity systems. Yeah, I don't really know the Uh-oh. Wait, is it happening? No, they're still liquid. Yeah, they have a I don't really know exactly what they have to deal with the regeneration issues, but they do have systems to deal with it. All right, we're going to hang out and just watch her for a while, I think. Ugh, unless she has enough water to go away. This is the thing where it's like, I don't want to poke the system too much because I don't want it to happen so quickly that we miss it. You were trying to convince him to buy a bento box. It's an all-in-one PCR, mini centrifuge, and gel dock entrance illuminator for 2,000. Wow. Yeah, I think I've heard of bento box before. It's pretty cool. Yep, she's still there. Are there any indicators, or is there any knowledge about how old they get? I'm sure there's information for specific species. I do not know this specific species. But yeah, Micro is correct. I do know that their lifespan is not as impressive as their ability to survive many years dehydrated. All right. I feel like, you know, if she stays put, we're in the end game now. I'm just gonna look around a little bit first. Oh, is she gonna go to the... Ugh, because there's a lot of water over there. 2 years is the max. Is that for like H. exemplaris, the model? Or is that just some random species? Yeah, Micro's our resident expert on most of these things. What you just saw was my tissue. You don't remember, it's just around the max. Fair enough, fair enough. Okay. I'm gonna keep going. I'm keeping on. I'm just rolling up little bits of tissue so that I can try and get this to go a little bit faster. Whoa. All right. The size of her body is starting to, oh, it's going to happen very quickly. So you can tell that the liquid is running very low because it's all kind of sticking to her. I'm just going to follow her around now. I'm not going to mess up. Yeah, she should start very soon. Oh, it's drying very, very quickly. And here we go. Once she stops being able to move, she will very quickly sort of like bunch up and dry out. And that was it. Believe it or not, that extremely, extremely quick transition is more or less the start of it. And I know that that's more or less the start of it because I've had them survive this. So that exact thing has happened. I've done it like 3 times so far, once on stream. You know, adding, it's just as violent every time. And we can come back, add water, and it will take a while. But unbelievably, that little like smudge will pop back into the shape of a tardigrade and then start moving around when she's ready. Yeah. So there we go. It's pretty awesome. All right. Now. Yeah, exactly. So as Micro is saying, in that short time, a bunch of really cool things are happening inside of her cells. It's an extremely controlled process, right? It has to be, more or less, for them to survive it. So for us, it just looks like they just completely desiccate. But protective proteins are being not assembled, but I thought it was like a glue or something like that. Maybe don't quote me on that. I need to read back up on my tardigrade cryptobiosis. But essentially they have to deal with the fact that they're these shearing forces and that proteins require water to maintain their shape, therefore function. Okay. So, you know, I normally stream until 4 my time. It is 4:40. I really appreciate the raid and the new follows. But I think it's about time for me to close up for the day. Green Herring, thank you for the follow. I appreciate it. Yeah, thank you for the clip. That's awesome. Ooh, they have intrinsically disordered proteins that fold into chaperones when water disappears. Awesome. Oh, yeah, thank you for linking the Discord. Yeah, I appreciate everyone new coming. And watching. Oh, Embus, you're here. I hope you were on for a little bit because we saw some cool stuff. And now I'm just about to leave. But that's okay. That's okay. I appreciate you stopping by anyways. Yeah, Discord's there if anyone wants to join. It's a pretty low, you know, not a ton of activity. So you probably won't get too much out of it, but it's where to go to post questions and pictures and stuff like that. Yeah, you'll just have to catch the VOD. Catch the last like 5 to 10 minutes. This is a tardigrade that we named that we have then dried out, but there's some cool stuff in there too. Thank you. When will I wake her back up? Probably when I wake the others up that are on this slide. I'm trying to collect some so I can get a nice little culture going. Maybe I'll do that. I actually don't know if I'll stream next Sunday because I'm kind of busy, but maybe I'll do that in like 2 weeks as I'll try to wake them all back up and see how they're doing. So let's find someone else to raid. You saw something you'd never seen before. A huge plus there. Well, awesome. I'm glad. Okay, so we raided Science. Whoa. Thank you for the bits. Right? Oh, 500. Wow. Sorry, the thing didn't show up too well for me. Thank you so much. Yeah, you'll just have to catch me next time. Yeah, I was just about to say, I think Volcano Doc. Science Streams is on too, but we just saw Science the other day. Yesterday, after the sausage making, not fiasco, but the sausage making adventure. So let's raid Volcano Doc. So I'm going to start that. Please stick around. So still science, but a bit of a shift. I see what is magma chamber simulator. That'll be interesting. All right, everybody. Thank you so much for sticking around, and I'll see you next time. Bye.